Assalamu alaikum. Revision of biology. Chapter 10.1 Structure of DNA. DNA is a polymer of nucleotides. So the name of the monomer that makes the DNA is the nucleotide. Each nucleotide consists of 3 parts, as you see. Number 1 in the middle is the 5 carbon sugar. 5C means 5 carbon atom sugar. This is oxygen. This is not 0. This is oxygen, and we count the carbon from here. 1, 2, 3, 4, 5, and carbon number 5 is outside the ring. To carbon number 1 attached a nitrogenous base or a nitrogen containing base, and to carbon number 5 attached phosphate group. The nitrogen base can be one of the 4: adenine, guanine, cytosine, or thymine. Adenine and guanine consist of 2 rings, so we call them double-ringed base like this. This is double-ringed, so this can be adenine or guanine. We have also single ring base like cytosine and thymine consist of 1 ring only, as you see here in the book. طبعا you should not you should not memorize the structure. I just want to show you that adenine and guanine consist of 2 rings. Cytosine and thymine consist of 1 ring. The single ringed base is called pyrimidine. The double ringed base is called biurine. Okay? Okay, type. Not necessary to memorize the term biurine and pyrimidine, but it is important to know that the bases can be double ringed or single ringed. Okay, so again, the nucleotide consists of 3 parts: deoxyribose sugar, as we are talking about the DNA, deoxyribose sugar, phosphate group, and nitrogen base. Phosphate group attached to carbon number 5, and nitrogen base attached to carbon number 1. Type. How the polymer is formed from the monomer. These are 2 monomers. This is the sugar, nitrogen base attached to carbon 1, phosphate group attached to carbon 5. Another nucleotide, okay, the sugar, the base, and the phosphate. We will make a dehydration reaction, in which dehydration means removal of water. So we will take the OH from carbon number 3 in 1 nucleotide, and we take hydrogen from the phosphate. So take OH from here, H from here, remove the water and make the bond. What is the type of this bond formed between the 2 nucleotides? Covalent bond. What is the name of the reaction? Dehydration reaction. Okay? Type. So now we joined the 2 nucleotides together by dehydration reaction. We took OH from here, we took H from here, and we joined it by making a covalent bond. Type. Continue this process, we will have a polymer of many nucleotides like we have here. Then, after forming the polymerization, let us talk about the Watson and the Crick model of DNA, or the double helix, and this is very, very important, the drawing and the description of the drawing. Okay, type. So here I have many nucleotides joined together as this way by dehydration reaction, and the formation of covalent bond. Type. So DNA is a polymer of nucleotide. Here they are. Each nucleotide consists of 5 carbon atom sugar, nitrogen base, attached to carbon number 1, and phosphate group attached to carbon number 5. 3. Polymerization takes place by dehydration reaction, as I explained here, where OH is taken from carbon number 3 here in 1 nucleotide, and the hydrogen from phosphate group in another nucleotide. I explained this. 4. DNA is double strand, because you know the story of Franklin when she was taking a picture for the DNA. She, as a chemist, when she can calculate the width of the DNA, how much it take a nitrogen base and a sugar and the phosphate, so she can calculate the width of the DNA strand. But every time she make the experiment and take a picture for the DNA and measure the width, she find that double its calculated value. And for example, if she calculated theoretically how should this width is, for example, X. And when she take a picture for the molecule and she measure the width, she has 2x. So she concluded that it is a double strand, X and X. So DNA is a double strand. Here it is, where the two strands are joined together by hydrogen bond. Okay, type. If this is a strand and this is a strand, so they are different molecule. They will be one molecule only when you join the two strands together. طب how the two strands will join together? By hydrogen bond. These dotted lines represent the hydrogen bond between the nitrogenous bases. Okay, so DNA is a double strand where the two strands join together by hydrogen bond between the complementary bases. 5. Yeah, make the two pages clear. 5. DNA is like a ladder. Here it is like a ladder where the sugar phosphate makes the backbone and the nitrogenous bases makes the rungs of the ladder. DNA is like a ladder where sugar phosphate is directed outward. As you see, this is to outside the molecule, and the nitrogenous base to the center or to inward, to inside the molecule. So again, let us read number 5. DNA is like a ladder where the sugar phosphate is directed outward and represents the backbone, and nitrogenous base are directed inward and represents the rungs of the ladder. Okay? Number 6. The base bearing rule. Adenine always join with thymine. Cytosine is always joins with guanine. So if I give you the nucleotide sequence in one strand, you can give me the nucleotide sequence in the other strand. Between A and the T, double hydrogen bond. Between C and G, triple hydrogen bond. As you see, triple. A and T, double bond. A and T, double bond. Okay? So the base bearing rule here, number 6. A joins with T, adenine joins with thymine by a double hydrogen bond. C binds with G, cytosine binds with guanine with a triple hydrogen bond. 7. DNA molecule is called anti-parallel. Anti-parallel. Why anti and why parallel? Anti means, as you see here, if one strand runs from 5' to 3' direction, the other strand will run from 3' to 5' direction. Aslan, what is the meaning of 5' and the 3'? Listen to this. This is the first nucleotide in the strand, and this is the last one. Here we have phosphate group free. يعني ايه free? Notice this phosphate group or are attached, are joined. But this phosphate group is free. It is not joined to anything. Okay, it is not joined to another nucleotide, right? So this is phosphate group. This free phosphate group, خلاص, attached to carbon number 1, number what in the sugar, to carbon number 5. This is why we call it 5' end. So again, 5' end means a free phosphate group, the end that has a free phosphate group attached to carbon number 5 in the sugar. Okay, similarly, this is called the 3' end because this is carbon number 3 and it has OH. This OH group is free. It is not taken in the bond as we did here. We took this OH in the bond, but this OH is free. This is OH is free. So this is the end that has free OH group attached to carbon number 3 in the sugar. This is why we call it 3' end. Okay, here you have OH, free OH attached to carbon number 3, so this is the 3' end. Here you have free phosphate group attached to carbon number 5, so this is a 5' end. So as you notice, one strand is opposite in respect to the other. This means it is anti-out. This is the meaning of the term anti. So anti means one strand is opposed to the other, or I can say it in another words. If one strand runs from 5' to 3', the other strand will run from 5' to 3'. Yani we don't have 5' and 5'. Or 3' and 3'. We have 5' and 5' opposite to each other. 3' and the 3' opposite to each other عشان واحد فيهم مقلوب بالنسبة للتاني. طب why this? يعني what is the importance of being one strand opposite to the other? بيقولوا إنه this makes the hydrogen bond stronger. يعني أنا لما أقلب واحد التي هتيجي بالظبط قبال التي، النيتروجين سبيس هتيجي بالظبط يعني in front of each other, so the hydrogen bond will be stronger. This will help to bind the nitrogenous bases يعني more strongly. This is why Allah inverted one strand in respect to the other. طيب what is the meaning of parallel؟ طيب طبعا you know better than me in math إنه when I say these are parallel strands means the width is the same. طب why the width is the same؟ because one nucleotide is single ringed, the other nucleotide is double. مفيش اتنين double قصاد بعض، مفيش اتنين single قصاد بعض. نرجع تاني للدروينج هنا. adenine binds with thiamine، ال A with T. طب الادينين double ring، الثيامين single، يبقى الويدث ايه؟ 3 rings، 2 و1. طيب جوانين always join with cytosine. This is double ring and this is a 1. يبقى 2 و1 يبقى الويدث 3. اوكي، وبالتالي دايما في البيز بيرنج واحده دبل رينج بتجوين مع واحده سنجل رينج، فالويدث دايما كونستانت اللي هو 3 3 3 3. لكن فور اكزامبل لو كانوا البيورين يعني الادينين مع الجوانين يبقى 4، الويدث 4، وهنا الويدث 2، هيبقى تويستد كده ان ايفن. 2 ده هيبقى الويدث قليل، 4 الويدث كبير، 2 الويدث قليل، 4 الويدث كبير and so on. مش هيبقى بارلل. لكن لما اخد واحد من هنا وواحد من هنا الويدث دايما تلاتات، فالويدث constant. This is why the backbone will come parallel to each other. I hope this is clear now. So here the DNA molecule number 7, I'm reading number 7. The DNA molecule is anti-parallel, means if one strand runs from 5' to 3' direction, here it is, the other strand will run from 3' to 5' direction. Why? Give reason for this. Why it is anti-parallel? This makes the hydrogen bond between the complementary bases stronger because it brings the nitrogenous base as close to each other. Number 8, the two strands are parallel to each other because the width of the molecule is constant. It's like a ladder. The width of the molecule is constant because one double ringed always join with one single ringed. Last thing, why it is parallel? Because in each base bearing, one base is single ringed, okay, and the other is double ringed, like A مع T double and single, C مع G single and double, so the width is 3's like this. Number 9 and the last one, DNA is helical shape. After the double strand is formed, it twists to right side like this, okay? So DNA is the helical shape as it is right hand twisted. Each turn consists of 10 base pair. يعني لو دي لفة كاملة لو عدينا البيز pair 1 2 3 4 5 6 7 8 9 10 يبقى كل لفة واخدة 10 pairs. اوكي، وبالتالي اللف ده بيقلل length of the molecule to 10th its length. This is the structure.